My name is Adam Williams. I'll be a junior at Benedictine High School in the fall. I'm in the Medical Laboratory Internship Program, and I currently work in the Microbiology Lab. My project is on the detection of herpes simplex virus, HSV, from patient samples using culture and molecular methods. Okay, so first we have a little bit of background about the information. Herpes simplex virus 1 and 2, HSV1 and HSV2, are members of the herpes virus family, Herpes viridae. HSV1 produces oral infections, as shown in the picture, while HSV2 produces genital infections. General characteristics of herpes viridae are as follows. They can develop a latent state after infection. During a latent state, the viruses can reside in the sensory ganglia located in the nervous system. They have a cytopathic effect on cells in which the cells are basically destroyed and become large bodies filled with cytoplasm and contain many nuclei. Finally, the viruses are held at bay by the cell-mediated immune system and can become reactivated by the weakening of this immune system, similar to AIDS. The current method used to detect herpes is ELVIS, enzyme-linked virus-inducible system. It is a more traditional cell culture method and is therefore considered the gold standard. Due to the required incubation period, however, the process can take between 16 and 24 hours. Furthermore, the results of the ELVIS method are subjective to the human eye under a microscope. Error can occur easily if a cell is overlooked. And the diagram at the bottom is a basic display of how the ELVIS system works. Scientists have engineered cells in which, when the cells are inoculated with a patient sample, if the patient sample contains the virus, the engineered cells will produce an enzyme known as beta-galactosidase. We stain for the beta-galactosidase, then it'll produce a blue result, which means the patient is positive, as shown in the picture at the far right. The test method in the experiment is the IsoAmp HSV assay, a rapid molecular test which amplifies the nucleic acids in order to detect the virus. Also, the test results are easily read. Similar to a pregnancy test, a positive or negative result is depicted by a red line. On the left, one red line is negative. On the right, two red lines is positive. So a comparison was made of both test methods, the ELVIS and the IsoAmp HSV assay. The hypothesis was that the IsoAmp HSV assay is both a faster and more sensitive molecular diagnostic test for the detection of HSV than the more traditional cell culture method of ELVIS. Because ELVIS is the method currently used in the lab to detect HSV, the 30 patient samples tested using the ELVIS method were leftover samples from the lab's previous day's work. The same 30 patient samples were then tested using the IsoAmp HSV assay. So the traditional cell culture method of ELVIS acted as a control in the experiment. On the right is a picture of a positive test result under the ELVIS method. On the left is an inverted microscope that we use to get the ELVIS results. Here's a simple diagram. We'll go into detail a little bit later in the presentation, but it basically shows the procedures for the IsoAmp. So the reagents and the controls are thawed. We take the viral transport medium, we take 25 microliters of that, put it in the dilution tube, centrifuge and vortex the dilution tube, and take 25 microliters of that into the reaction tube. A master mix is added into the reaction tube, which consists of an enzyme amplification reagent. We put the reaction tube in the heating block for an hour at a temperature of 64 degrees centigrade. Reagent preparation. Amplification reagent tubes, positive and negative controls, were thawed at room temperature prior to being centrifuged and used. Specimen preparation. 25 microliters of the viral transport medium were transferred from each collection vial or control tube to the corresponding 2.0 dilution tube pre-filled with dilution buffer. That's what's on the left, being the viral transport being transferred from the viral transport medium into the dilution tube. After centrifuging the dilution tubes, 25 microliters are transferred from the dilution tubes to the corresponding 0.2 milliliter reaction tubes, which is the picture on the right. Amplification. Master mix was prepared by transferring 25 microliters of the enzyme reagent to the amplification reagent tubes. That's the picture on the left. That's the master mix being prepared. 25 microliters of this solution was put into the reaction tubes. Finally, 2 or 3 drops of mineral oil were added to the reaction tubes to prevent condensation. The reaction tubes were then put into the heating block for an hour at a temperature of 64 degrees centigrade. Detection. After assembling the Amplicon cartridge with the reaction tube, the closed Amplicon cartridge was inserted into the detection chamber. The results were read after 15 minutes. That's the two pictures shown here of how to assemble the Amplicon cartridge. Both the IsoAmp HSV assay and the ELVIS assay produce equal positive and negative test results, resulting in equivalent sensitivity and specificity, as shown here. Conclusions. Because the two test methods produced equal results, the hypothesis which stated the IsoAmp HSV assay was more sensitive was unable to be confirmed at this time. Although the IsoAmp HSV assay and ELVIS have comparable sensitivity, the IsoAmp HSV assay is more beneficial for the clinical setting, especially in smaller facilities, because it has the same sensitivity as the traditional cell culture method, but it gives a faster result. Even though the IsoAmp HSV assay is undoubtedly the faster diagnostic test for identifying HSV, the IsoAmp test will also detect herpes, dead or alive, which produces a positive test result even if the patient no longer has the live herpes virus. I would like to give a special thanks to Rosalind Strickland. She is a Senior Director of the Cleveland Clinic Office of Civic Education Initiatives. Nedra Starling is also a member of the Office of Civic Education Initiatives. My mentor, Dr. Yen, who provided me with the topic of my presentation. My co-mentor, Deborah Cohn, she walked me through the procedures of my project. And finally, the BioHelix Corporation, who provided all the free supplies, reagents, etc. Thank you.